All right, so we're going on another deep dive today, and you sent this fascinating biology chapter about gene regulation, and it just makes you realize it's incredible that every single cell in our bodies has the same DNA,
That's right.
but they can become so specialized, like a skin cell, a muscle cell, a brain cell.
Yeah,
It's like they're reading different chapters of the same instruction manual.
it really is. It's like having one huge cookbook with all the recipes in it, but each cell only chooses to cook a specific dish depending on its role in the body.
Well, now I'm hungry, but okay, that's a great analogy.
hmm
So how do they decide which genes to express, right? Because the chapter starts with this really cool example of the quattro ojos fish.
Yes, the quattro ojos fish. So this fish has evolved this remarkable adaptation where it's got these eyes that are actually split, and the upper portion is adapted for vision in air, and the lower portion is adapted for underwater vision.
So it's like having built-in goggles.
Built-in goggles, that's exactly right. And what's fascinating is the cells in both parts of the eye, they contain the same genes for vision, but they express them differently. So if you look at figure 18.1 in the chapter, it actually shows how the genes for aerial vision are more active in the upper part of the eye, while the genes for aquatic vision are more active in the lower part.
So even though all the cells have the same DNA blueprint, they're using different parts of it. It's like having a toolbox with all the tools, but each cell only picks out the ones it needs for its specific job.
That's a really good way to think about it.
Right.
Now let's shift gears a little bit and talk about bacteria, which are these single-celled organisms, right? And they regulate their genes in a very efficient way using these things called operons.
Operons, what are those?
So imagine a factory assembly line that produces a specific product. An operon is like a set of genes that work together to make that product, and they're all under the control of one on-off switch called an operator.
So it's like a coordinated team of genes all working together.
Exactly like a coordinated team, and the textbook gives a great example of the TREP operon, which controls the production of tryptophan. So when tryptophan levels are low, the operon is switched on, and the bacteria can start making their own tryptophan. But when tryptophan is already available, it actually binds to this repressor protein, and it activates it.
Wait, so the tryptophan itself helps turn off its own production.
Precisely. It's like a feedback loop. And this activated repressor then binds to the operator, switching the operon off. And if you look at figures 18.2 and 18.3, it illustrates this mechanism beautifully. It's a really efficient system because it ensures that the bacteria are only producing tryptophan when they actually need it, so it saves energy and resources.
That's incredibly efficient. It's like a self-regulating system.
It is a self-regulating system, and bacteria have other types of operons too, like the lac operon is another great example, but instead of being repressible, like the trip operon, the lac operon is inducible.
Inducible, what does that mean?
It means that the operon is usually off, but it can be turned on in the presence of a specific molecule, and in this case, it's lactose, which is a sugar found in milk.
Ah, so the bacteria can switch on the genes to digest lactose only when there's milk around.
That's exactly right. So the presence of lactose triggers this whole series of events that ultimately removes the repressor from the operator, and this allows the genes for lactose metabolism to be expressed. Figure 18.4 shows this process in detail.
Wow, bacteria are so smart.
Yeah.
So we've seen how bacteria use these operons for really efficient gene regulation, but how do things work in more complex organisms like us?
Well, eukaryotic gene regulation is a lot more intricate, for one thing. Our DNA is packaged with proteins to form this thing called chromatin, and chromatin structure can actually influence which genes are accessible for transcription.
So it's not just about the genes themselves, but also how they're packaged.
Think of it like this. If you have a library full of books, the way those books are organized can affect how easily you can find the information you need. And similarly, the structure of chromatin can determine which genes are readily available to be transcribed.
Okay, so the packaging itself is a form of regulation.
It is.
And the chapter mentions these two types of euchromatin and heterochromatin.
That's right. So euchromatin is more loosely packed, and this makes the genes within it more accessible. Heterochromatin, on the other hand, is really tightly condensed, so it makes it difficult for the cellular machinery to actually access the genes.
So it's like having some books on easily accessible shelves and others locked away in a vault.
Mm-hmm.
Interesting. And then the chapter also dives into this concept of epigenetics, which sounds really fascinating.
Right.
What exactly is epigenetics?
So epigenetics refers to changes in gene expression that occur without alterations to the underlying DNA sequence itself. So it's like adding bookmarks or highlighting certain passages in that library book. You're not changing the words themselves, but you're altering how they're read.
I see. So it's like a layer of control on top of the DNA sequence itself. What are some of the mechanisms that are involved in this?
So two key players are histone modifications, and DNA methylation. Histones are the proteins that DNA wraps around, and their tails can be chemically modified, and this influences how tightly the DNA is packaged.
So it's like adding or removing decorations on the histones to signal whether a gene should be expressed or not.
Exactly. And these modifications can actually be passed down from one cell to its daughter cells, influencing gene expression patterns in future generations of cells. And figure 18.7 gives you a nice visual representation of this concept.
Wow, that's pretty remarkable. And what about DNA methylation? How does that work?
So DNA methylation involves attaching a methyl group directly to the DNA molecule, usually on a cytosine base, and this often acts as a signal to repress gene expression.
So it's like putting a do not disturb sign on a gene.
That's a good analogy, and the really interesting part is that these epigenetic changes can be influenced by environmental factors.
But you mean like our environment can actually affect how our genes are expressed?
There's growing evidence to support that idea, and the chapter highlights these fascinating studies on agouti mice, where the mother's diet during pregnancy actually affected the coat color and the health of her offspring.
So what a mother eats can actually influence the expression of genes in her babies. That's amazing.
It is.
And the chapter also mentions the Dutch hunger winter, right?
Yes, that's another compelling example. Children who were born to mothers who experienced famine during pregnancy, they showed long-term health effects, and these effects were linked to epigenetic changes that occurred during fetal development. Figure 18.8 shows some real-world examples of these epigenetic influences.
This is incredible. It means that our experiences, and even the experiences of our parents, can leave this lasting imprint on our genes.
It really challenges this traditional view of nature versus nurture. You know, we're starting to understand that it's not a simple either-or proposition. There's this complex interplay between our genes and our environment.
This is mind-blowing. So we've talked about chromatin structure and epigenetics as ways to regulate gene expression. But what about the genes themselves? The chapter mentions these things called transcription factors, and I'm curious to learn more about them.
So transcription factors are like the conductors of the gene expression orchestra. They're proteins that bind to specific DNA sequences, and they control the rate of transcription.
So they decide how much of a particular protein is produced.
Precisely, they can act as activators boosting transcription, or they can act as repressors hindering it.
It sounds like a very precise control system, and the chapter mentions that there are different types of transcription factors.
Yes, there are general transcription factors that are essential for the transcription of all genes, and then there are specific transcription factors that regulate the expression of particular genes.
So the general ones are like the foundation, and the specific ones are kind of fine-tuning the expression of individual genes.
That's a good way to put it, and these specific transcription factors often bind to regions called enhancers, which can be located far away from the gene they regulate.
Wait, how can something so far away control a gene?
Well, the DNA can loop and bend, bringing the enhancer and its bound transcription factors close to the gene's promoter region, and figure 18.9 shows how this works.
It's like having a remote control that can operate from across the room.
That's a good analogy. And these enhancers, along with their specific combinations of transcription factors, they act like these control panels. They fine-tune the expression of individual genes.
This is all so complex, but incredibly fascinating. It's amazing to think about all these intricate mechanisms that are happening inside our cells right now to control which genes are expressed and how much protein is produced.
It truly is a remarkable system, and we've only just begun to scratch the surface, but we're learning more all the time, and these discoveries are revolutionizing our understanding of biology and medicine.
Yeah, really amazing stuff.
It is.
All right, well, let's take a quick break and we'll come back and we'll keep going on this deep dive into gene regulation.
Sounds good. And this whole intricate dance of gene regulation, it doesn't just stop with transcription.
You mean there's even more control after the initial RNA message is made?
Absolutely. There are multiple checkpoints along the way to make sure that gene expression is fine-tuned at every level. And the chapter dives into some really fascinating mechanisms of what's called post-transcriptional regulation.
Post-transcriptional regulation, it sounds like quality control for gene expression.
Exactly. The cell has several ways to kind of tweak the message even after that initial RNA transcript is made. And one key mechanism is alternative RNA splicing.
Alternative splicing, it sounds kind of like editing a film. You know, you can take the same footage and create different versions of the movie depending on how you cut it.
That's a brilliant analogy. That's exactly what it's like. So with alternative RNA splicing, the cell can choose which segments of the RNA molecule to keep and which ones to remove. And this results in different protein products from the same gene.
So one gene can actually code for multiple proteins. That's incredibly efficient.
It is incredibly efficient, and the chapter gives this example of the troponin T gene, which can be spliced in different ways to produce slightly different forms of the protein that's involved in muscle contraction.
So even subtle variations in this splicing can have significant effects on protein function. It's like adjusting the recipe slightly to create different flavors of the same dish.
Precisely. And the cell can also regulate the initiation of translation, how efficiently the ribosome binds to the mRNA and starts making the protein.
So it's like controlling the speed at which the recipe is being cooked.
Exactly. And it can also control the lifespan of the mRNA molecule itself. Some mRNAs are degraded very rapidly, while others hang around for days or even weeks.
So it's like adjusting the shelf life of those protein blueprints. Fascinating. And what about after the protein is made? Can gene regulation still play a role at that point?
Absolutely. Even after a protein is synthesized, the cell can modify it in various ways, activating it, inactivating it, or even sending it to specific locations within the cell. And ultimately, the cell can also regulate how long a protein survives before it's degraded and recycled.
So gene regulation is involved from start to finish, from that initial DNA blueprint to the final protein product and everything in between.
It's a remarkably orchestrated process, and we're still unraveling all of its complexities. But one of the most exciting areas of research in recent years has been the discovery of these things called non-coding RNAs.
Non-coding RNAs. I remember reading about those. They're like the dark matter of the genome,
Yeah.
right? You used to think they were junk DNA, but now we're realizing they have all these important functions.
Exactly. For a long time, scientists were really focused mainly on those protein coding genes. But it turns out that a vast majority of our DNA is actually transcribed into these non-coding RNAs, which play a wide variety of roles in regulating gene expression.
So they're like these hidden regulators pulling the strings behind the scenes.
That's a good way to think about it. And one of the most fascinating types of these non-coding RNAs are these things called microRNAs or miRNAs.
Immunase, I've heard about those. They're tiny but powerful, right?
They are indeed. miRNAs are like tiny assassins. They can bind to complementary sequences in those mRNA molecules and either trigger their degradation or block their translation, essentially silencing the message.
Wow, so they can target specific mRNAs and prevent them from being turned into proteins. That's incredible. And the chapter mentions that miRNAs might regulate the expression of as much as half of all human genes.
That's right. And they're emerging as key players in a wide range of cellular processes, including development differentiation and even diseases like cancer.
So these tiny RNAs are having this major impact. It's like discovering a whole new layer of control in the cell.
Absolutely. And there are other types of non-coding RNAs too, like small interfering RNAs or siRNAs, which are similar to miRNAs in their ability to silence genes. And then there are these longer non-coding RNAs or LNC-RNAs, which can regulate gene expression in various ways, including modifying chromatin structure and acting as scaffolds to bring together different protein complexes.
So non-coding RNAs are really adding this whole new dimension to our understanding of gene regulation. It's like we're just beginning to appreciate the full complexity of the genome.
It's a rapidly evolving field, and there's still so much we don't know. But one thing is clear, these non-coding RNAs are essential players in this intricate dance of gene expression.
This is also fascinating, but I'm also curious to see how these principles of gene regulation apply to something as complex as embryonic development. Like, how does a single fertilized egg give rise to all the different cell types in an organism?
That's a question that has captivated biologists for centuries. And thanks to advances in molecular biology, we're starting to really unravel the mysteries of embryonic development.
And the chapter uses the example of frog development, right? Like, how do these amphibians transform from a single cell into a tadpole and eventually a frog?
It's a remarkable process orchestrated by this precise program of gene regulation, and the initial instructions are provided by these cytoplasmic determinants, which are these maternal substances in the egg that influence the expression of genes in that early embryo.
So it's like the mother is packing a suitcase full of instructions for the developing embryo.
That's a great analogy, and as the embryo divides, the cells communicate with each other through these signaling molecules, triggering changes in gene expression that guide their differentiation into specific cell types.
So it's a combination of these pre-programmed instructions and then this ongoing communication that shapes the developing organism.
Precisely. And the chapter highlights this concept of induction, where one group of cells can influence the development of another group through these signaling molecules.
Yeah, it's like this chain reaction where one signal triggers another and another, ultimately leading to the formation of different tissues and organs.
Exactly, and the chapter focuses on muscle cell differentiation as a case study to illustrate this process.
Muscle cells, those are pretty important for, well, moving around and doing all sorts of things. How do they develop?
So muscle cells, they arise from these embryonic precursor cells that have the potential to become various cell types, but they receive signals that commit them to the muscle cell fate. And this commitment is called determination.
So it's like choosing a career path. Once you're determined to be a muscle cell, there's no turning back.
Exactly. And once a cell is determined, it undergoes differentiation, expressing the genes that give it that characteristic structure and function of a muscle cell. And this process is controlled by these master regulatory genes.
Master regulatory genes, those sound pretty powerful.
The chapter focuses on one called MyoD, and this gene encodes a transcription factor that activates the expression of various muscle-specific genes, including genes for those contractile proteins, actin and myosin.
So MyoD is like the foreman on a construction site directing the building of a muscle cell.
That's a great way to visualize it. And MyoD even activates its own expression, ensuring that that muscle cell fate is maintained.
Amazing. So we've seen how gene regulation guides cell differentiation, but how does the organism as a whole take shape? The chapter mentioned something called pattern formation.
Yes, so pattern formation is the process by which the overall body plan of an organism is established. It's like creating a blueprint for the building, where the head goes, where the limbs go, and so on.
So it's all about this spatial organization. And the chapter uses the fruit fly Drosophila as an example. Why Drosophila?
So Drosophila has been this model organism for developmental biology for over a century. It's relatively easy to study in the lab, and its development is surprisingly similar to that of more complex animals, including humans.
So what have we learned about pattern formation from studying Drosophila?
One of the key discoveries was the role of these maternal effect genes, and these genes are expressed in the mother, and their products are deposited in the egg, providing that initial positional information for the developing embryo.
So it's like the mother is giving the embryo a map to guide its development.
Exactly, and a classic example is the bicoid gene mutations in this gene result in embryos that have two tails and no head.
So they knew bicoid was somehow involved in specifying the head end.
Right, and through a series of elegant experiments, they tracked down the bicoid mRNA and its protein product, and they discovered that the mRNA was localized at the anterior end of the egg.
So that's how the embryo knows which end is up. The bicoid mRNA is like a signpost marking the head territory.
Precisely. And after fertilization, the bicoid mRNA is translated into protein, which diffuses from the anterior end towards the posterior, creating a concentration gradient.
So the highest concentration of bicoid protein signals head, while lower concentrations mean more towards the tail. It's like a chemical coordinate system.
That's a great way to visualize it. And this bicoid gradient, it acts as a morphogen, providing positional information to the developing embryo.
And to confirm its role, researchers injected bicoid mRNA into different regions of mutant embryos, and guess what happened?
Well, they were able to induce head formation at the injection sites, proving that bicoid was indeed the master regulator of head development.
That's incredible. It's like reprogramming the embryo's destiny by manipulating a single gene.
It's a testament to the power of genetic analysis and the ingenuity of scientists in unraveling the mysteries of development.
It also highlights this fundamental role of maternal effect genes in laying the foundation for the embryo's body plan.
And as development proceeds, the embryo's own genes come into play, refining the pattern that was established by those initial maternal cues.
It's like a symphony of gene regulation with different sets of genes taking the stage at precise times, guided by this complex interplay of signals and transcription factors.
And while Drosophila provides this captivating glimpse into these processes, the principles that we've discovered in these flies have far-reaching implications for understanding development in all animals, including humans.
It's a reminder that even the most complex organisms share this common evolutionary heritage, and the study of simpler model systems can illuminate the secrets of our own biology.
And as we delve further into the chapter, we'll see how these same mechanisms of gene regulation that orchestrate development are also implicated in diseases like cancer.
Cancer, a disease of uncontrolled cell growth, it often arises from disruptions in that delicate balance of gene expression that normally keeps cell division in check.
And the genes involved in cancer are often the same ones that play these crucial roles in embryonic development, highlighting these intricate connections between these fundamental processes.
And as we've learned, cancer is rarely caused by a single mutation. It's typically a multi-step process with multiple genetic hits accumulating over time.
The chapter delves into the specific genes and pathways involved in this progression, focusing on two key players: oncogenes, and tumor suppressor genes.
So oncogenes are kind of like the gas pedal stuck down, pushing the cell to divide uncontrollably. And then the tumor suppressor genes are like the brakes, trying to keep things under control.
That's a great analogy, and mutations in either of these types of genes can really disrupt that delicate balance of cell division, and that can lead to cancer development.
And the chapter dives into some specific examples of these, right? Like the RAS proto-oncogene and the P53 tumor suppressor gene.
Yes, those are two of the most well-studied genes that are involved in cancer. So the RAS gene encodes a protein that helps relay signals from these growth factors outside the cell to the nucleus, and that ultimately promotes cell division.
So it's like a messenger that tells the cell when to divide.
Exactly. But mutations in the RAS gene can create this hyperactive protein that's sending those signals constantly, even when there are no growth factors around, and this leads to uncontrolled cell division.
It's like a messenger that just keeps shouting, divide, divide, even when nobody's talking.
Exactly. And then there's P53, which is really a remarkable gene. It acts as a guardian of the genome.
A guardian of the genome, it sounds pretty important.
It is. It's a tumor suppressor gene that plays this critical role in detecting and responding to DNA damage.
So it's like a quality control inspector, making sure everything is in order before the cell divides.
Exactly. If P53 detects damage, it can halt the cell cycle, giving the cell time to repair the damage. It can even trigger this programmed cell death, or apoptosis, if the damage is too severe, preventing that damaged cell from dividing and potentially becoming cancerous.
Wow, so P53 is like a superhero protecting us from cancer.
In a way it is, but sadly, P53 itself is often a target of mutations in cancer. More than half of all human cancers have mutations in the P53 gene, disabling this crucial safeguard.
That's kind of sobering, but the chapter also points out that cancer development is usually a multi-step process.
Right.
It's not just one mutation that suddenly leads to a full-blown tumor.
Right. It usually takes multiple mutations that accumulate over time to transform a normal cell into a cancerous one. And the chapter uses colorectal cancer as a really good example of
That
this.
colorectal cancer, that's one of the most common cancers, right? So understanding how it develops is really important.
It is, absolutely. And colorectal cancer often starts with this small, benign growth called a polyp, and that polyp can gradually accumulate mutations over time.
So it's like a series of unfortunate events, each mutation kind of pushing the cell further down that path towards cancer.
And the chapter highlights the importance of early detection and screening for colorectal cancer because those polyps can often be removed before they become cancerous.
That's why colonoscopies are so important, right? They can catch those polyps early on, preventing them from progressing.
Exactly. And understanding the genetic basis of cancer has also led to the development of these targeted therapies that specifically attack cancer cells while sparing healthy cells.
That's amazing. So knowledge really is power when it comes to fighting cancer.
It really is. And the chapter also discusses the role of inherited predisposition in cancer development.
So some people are genetically more likely to develop certain cancers than others.
Yes, some individuals inherit mutations in genes that increase their risk of developing specific cancers. For example, mutations in the BRCA1 and BRCA2 genes are linked to an increased risk of breast and ovarian cancer.
So knowing your family history and getting genetic testing, if it's appropriate, can be really important for early detection and prevention.
It can empower individuals to make more informed decisions about their health. And finally, the chapter touches on this fascinating role of viruses in cancer development.
Viruses, those tiny infectious agents that can make us sick, right? But they can also cause cancer.
Indeed, some viruses can insert their genetic material into the host cell's DNA, and this can disrupt gene regulation and potentially lead to cancer.
It's like a hostile takeover of the cell's genetic machinery.
In a way, it is, and the chapter highlights the example of the human papillomavirus, or HPV, which is a major risk factor for cervical cancer.
And luckily, we now have vaccines that can protect against HPV infection.
Yes, that's a huge breakthrough in cancer prevention. It shows how understanding these viral mechanisms of cancer can lead to really effective strategies for protecting our health.
Well, this deep dive into gene regulation has been truly eye-opening. It's amazing to see how this one complex process underlies so many aspects of life, from embryonic development to the onset of disease.
It really is a testament to the power of scientific inquiry and the ingenuity of researchers who have dedicated their lives to unraveling these mysteries.
It really makes you realize that even though we've learned so much, there's still so much more to discover.
Absolutely. Gene regulation is a very dynamic and ever-evolving field, and each new discovery brings us closer to understanding the fundamental principles of life.
Well, on that note, I think it's time to wrap up this deep dive into gene regulation. I feel like I've learned so much, but it's also sparked a whole new set of questions in my mind.
And that's the beauty of science. It's a journey of continuous exploration and discovery.
Well, thank you so much for being such a fantastic guide on this journey. I'm sure our listeners feel just as enlightened as I do.
It was my pleasure. And remember, the world of gene regulation is full of wonders, so keep exploring, keep questioning, and keep that curiosity alive.
And to all our listeners, thanks for joining us on the Deep Dive. We'll be back next week with another fascinating topic to explore.
Until then, keep learning and stay curious.